We're going to move on to our last but not least presenter of the day, Amanda Nurse, and she's from the University of Western Ontario, and she's a scientist looking at salmon populations in the Great Lakes. So take it away, Amanda.
Hi, thanks for that introduction. So I'm Amanda. Yeah, like Marianne was just saying, I just graduated from an undergraduate degree in biology at Western University. I did a thesis project in Dr. Brian Neff's lab on the utility of nuclear versus mitochondrial environmental DNA for population monitoring in Atlantic salmon. So there's just a little bit of a lag here. Oh. Okay, so first to start off, what is environmental DNA? So it's DNA that is shed from an organism as it interacts with its environment. There are studies that look at environmental DNA terrestrially, so you can look at sediments or samples from a tree if an animal has brushed up against it. But for me, I was looking at the aquatic side of that in fish species. So it can be shed cells in the form of hair, skin, scales, or remnants. For the majority with the fish that I was looking at, it's mostly shed scales, and there's many factors that can influence the amount of DNA that you might find in a set body of water. So things like the temperature, the pH, and the salinity of the water can influence both the rate of shedding of those scales that contain the DNA, as well as the degradation of the DNA once it's in the water column. So how is it used in biology? So this is an image that Stephanie actually took for the River Institute. They have been taking some environmental DNA samples. So the first step is to collect those water samples. Here you can see you use an extended arm just so that you're not stirring up the water close to the locations, so that you don't get a ton of sand particles or anything else to contaminate your sample. And then you run that water sample through a filtration apparatus. So I used a nitrocellulose filter membrane, which traps all the DNA on that membrane. And then you can cut up that little piece of filter paper and do an extraction protocol. So for mine, I just used a Qiagen kit, and you would do it the same as you would for DNA extraction from any other tissue. So I also was taking fin clips and doing the DNA extractions just as a positive control to confirm that I was getting the amplification of the DNA from that water sample. So once you have that DNA extracted from the filter paper, you can amplify it using PCR, and you use primers specific to the species that you're looking at. So specifically for my experiment, I had ordered some Atlantic salmon primers for my PCR, and then you can detect whether the Atlantic salmon are present in that body of water or absent. So I was just going to go over first why we would use environmental DNA monitoring techniques and some of the more traditional approaches. So more traditional approaches include electrofishing, seine netting, also gill netting. So these kind of practices are very beneficial and helpful if you're looking for information regarding the individual sizes of the fish that you're looking at. So you can get length measurements, biomass, a lot of these different characteristics. But the downside to using this type of method is that it tends to be very invasive and stress-inducing. There were a lot of studies showing that the cortisol levels in fish increased quite a bit after handling, which is just an indicator of stress, specifically in Atlantic salmon as well. So it's also difficult for small population abundances. So you can imagine if you only have a very small population in a set body of water, it's first of all a little bit more difficult to actually capture that small group of individuals. And you also don't want to be inducing more stress or causing anything that could increase the mortality of that population if they're already at very low numbers. It also tends to be a little bit more time intensive just to train researchers to actually go out and do these netting techniques, and also identifying the fish species as you obtain them. So an environmental DNA approach offers no direct interaction with the focal individuals. There's no added stress on the fish because you're not actually directly interacting with them. You just need a water sample. And it's really good for early detection of invasive and rare species. So it's first of all really easy to just train someone to go out and grab a water sample, and then it can be sent off to lab. And it's also, yeah, you're not actually directly in contact with any of the fish species that you're looking at. And for my experiment specifically, from the point whenever I took the water sample to when I got my DNA results of whether there was a species present or absent, it only took 2 days at a maximum. That was with an overnight incubation, so it's a very quick indicator where you can determine what species are present in the body of water that you're looking at. And more specifically, with environmental DNA, up until now, researchers have been using mitochondrial markers. So each cell has a high copy number of mitochondria, so that means that there's a lot more mitochondrial DNA within each cell, as well as within the water sample that you're taking. So it's a little bit more reliable because you do have so much DNA contained in those samples whenever you're looking at the mitochondrial markers. But the caveat to using that is that they're non-specific to individuals. So you can only get an assay of the presence or absence of a species. There's not much more information that's provided. So that's what I wanted to mainly look at in my experiment, was using nuclear markers, which are less abundant in sample. However, they can have individual specific alleles, and you could obtain information on the number of individuals present. So I use the mitochondrial markers kind of as a control for this experiment because it has been done before, but nuclear markers is kind of new territory where we're trying to see if we could get those to amplify as well. So specifically for the mitochondrial marker that I used the gene cytochrome oxidase subunit 1, which is actually the gene that encodes the terminal enzyme in the electron transport chain. So it's very highly conserved among species, and there's actually a short barcoding region near the beginning of the gene. So this region is species specific. So I ordered primers that were specific to the Atlantic salmon barcoding region for that gene. And that way we know that whenever we're amplifying the DNA, we're only amplifying Atlantic salmon DNA from that segment of the genome. So for my nuclear marker that I use, I looked at microsatellites, which are sections of repetitive DNA. They tend to have a very high mutation rate. They have a lot of insertions or deletions of the entire repetition of DNA. So it's a lot more a lot higher genetic diversity. So it would just give information about the number of alleles present, and then using that, we could extrapolate the number of individuals that are present in a body of water. So specifically why I was looking at Atlantic salmon for this study is that they are currently endangered in North America. And this graph is just showing the decline of the Atlantic salmon from about 1970 to 2016. And as you can see, in the early 1990s, the population size actually fell below the conservation minimum, which is just the minimum number of Atlantic salmon that are required in order to maintain a sustainable population. So it's important to be able to monitor these species and not use invasive approaches that could increase the mortality rates of these species if you're directly interacting with them. And another reason why environmental DNA could be helpful in this area is for reintroduction efforts. So specifically in Lake Ontario, in the late 1800s, there was a lot of habitat destruction, and it drove the Atlantic salmon population to local extinction in that area. And unfortunately, despite Lake Ontario recovering in terms of its health conditions, reintroduction efforts have failed, which is surprising. So they've tried to reintroduce the Atlantic salmon to this body of water, but despite it being theoretically a habitat that could sustain their population, they haven't for some reason. So one potential reason that researchers have theorized for this being the case is the replacement of their prey species that the Atlantic salmon originally preyed upon by a new exotic species that wasn't there before. So the Atlantic salmon just haven't adapted to this new species that's replaced the original one in the food chain. So an environmental DNA monitoring approach can just contribute to the solution, where it could monitor the population sizes of Atlantic salmon, but also look at many different species, specifically if exotic species are moving into an area. And that way we can better determine where we should put our reintroduction efforts and decide where it would be worthwhile, as opposed to in Lake Ontario, it hasn't been successful. Sorry, there's a bit of a lag with the slides. Okay, I think think I jumped there. So specifically for my experiment, the aim was to develop and validate a method of Atlantic salmon detection using microsatellites obtained from an eDNA sample. So I had two main objectives. The first was to design methods that confirm the use of microsatellite markers to detect the presence of Atlantic salmon. And secondly, I wanted to recover multiple individual specific alleles from a single eDNA water sample to confirm methods of individual specific detection. So the population that I used, there's a bit of a lag again, there we go. The population that I was using of Atlantic salmon were actually originally from the LaHave River in Nova Scotia, and they were obtained as fertilized fish eggs from the Normandale Fish Culturing Station here in Ontario. At Western, we have a fish hatchery with many Atlantic salmon to choose from, so those are the ones that I used for my experiment, and everything for my experiment was done in the lab. There is a master's student this year who's taking over and going to try some field experiments with this. So the methods that I used, I put either one Atlantic salmon individual or two in a laundry basin where I covered off the drain and added 15 liters of water. And I let those Atlantic salmon swim around for 24 hours before collecting 1 liter of water sample. And then that water sample is run through a vacuum pump filtration apparatus. So as you can see here, water is poured into the top of this apparatus. And then that nitrocellulose filter paper that I was talking about earlier is seated in the middle. And there's a vacuum pump hose that's attached to the bottom, which allows for all of the larger particles to be captured on top of the membrane, and any smaller particles will be filtered through so that filter paper is just saturated with all of the DNA that you're looking for in your experiment. And after the sampling of the water for each trial that I conducted, I also took a fin clip just so that I could match up the water sample DNA to those fin clips kind of as a positive control to make sure that my amplification was working well. So once I had that filter paper and the fin clips, I did a DNA extraction. I just used a Qiagen kit. And then I conducted PCR amplification using either primers for the cytochrome oxidase subunit 1 gene, which is the mitochondrial one, or using the microsatellites. And the last step was going to be an ABI fragment analysis. So the DNA that I had amplified using PCR amplification, I was just going to send that off to get analyzed just to see the different alleles that we were getting using the microsatellites. Unfortunately, whenever I was conducting this project, it was right when COVID was starting up, and the fragment analysis lab actually closed down at that time, so I wasn't able to complete just this last step, but yeah, they should be able to. They're going to continue this project and see if they can get that information to us shortly. So these are just some of the results. Here I just wanted to show the amount of DNA contained in either the fin clip samples versus the water samples. So here the darker gray bars are the nucleic acid content contained in the fin clip samples. And as you can see, it was a much larger amount compared to the water samples. So it is very important to make sure that your DNA extraction protocols are very specific and there's no contamination because it's already a very small amount that you're working with. One interesting thing that I found was there was no difference in the amount of nucleic acid contained in the water samples using 1 fish in the container compared to 2 fish. And this was actually shown in a previous study where the amount of eDNA in a set body of water actually reaches a point of saturation where it doesn't increase anymore after about 16 hours. So I'm assuming that this was the cause for the not having an increase in the amount of DNA here. And these are the results from the PCR amplifications of the DNA using the cytochrome oxidase subunit 1 genes. So that's the CO1 that you see up here. The numbers next to them just represent different primers that I was using. I was just doing some optimization to see which ones would work best. So the first 2 lanes in either sequence is the amplification of DNA from the fin clips. So they all had strong amplification. And then lanes 3 and 4 were the amplification of the water samples. So again, I did get pretty strong amplification for the majority of these. So just showing that it was pretty reliable to use the mitochondrial DNA whenever extracting that from the water samples. And going forward, I used this mitochondrial marker to represent just a positive control to confirm whenever I was looking for my microsatellite primers or microsatellite genes. So here was just the results from the experiment whenever I had one fish in the container. So for this one, again, I just used the cytochrome oxidase subunit 1 as the positive control. And then SSSP was the microsatellite genes that I was looking for. And again, it was just 3 different primers that I sampled that with. So for this one, the first 3 lanes in each sequence was using fin clip DNA, and lanes 4 to 6 was using the water samples. So again, my cytochrome oxidase subunit 1 worked very well. And then the amplification of the fin clips worked well with the microsatellites. I did get less amplification for the water samples, but lane 6 tended to show pretty strong amplification. So it's at least a good first step, and confirming that this can be used. There might be a few more, a little bit more refinement in being able to amplify it and visualize it a little more effectively on the gels, but there was strong indication that it was working in at least some of the trials that I was using. And then again, this was just using the 2 fish in the containers, and here I just circled a few of the columns where I was amplifying water samples, so you can see that there was some clear amplification for that one as well. So just the significance of this whole experiment once it's finally completed and the field trials are done, if methods are developed that can reliably extract and amplify these nuclear microsatellite DNA obtained from the water samples, we'll be able to get estimates of the number of individuals in a population. And we can also look at the allelic frequencies and within population genetic variation, as opposed to what we were looking at before with just the presence or absence using mitochondrial. So I'd love to take any questions if anyone has some.
Thank you so much, Amanda. That was great. We do have a couple minutes for questions, and we have a question that came in here. Emily asked, well, she said, great presentation and explanation of eDNA. Usually an easy thing to explain, so you did a fantastic job on that. Have you heard of any discussions about whether eDNA could replace active and passive methods of monitoring fish populations, or does catching fish maybe complement eDNA for a more complete data set?
I think people are gradually starting to switch over to using eDNA. There has been some discussion of how reliable it actually is, and how soon after, for example, a fish has moved away from a certain area where you're sampling from, that it can still reliably be detected. So there's still a lot of questions, but hopefully if we're able to answer and find out some of these variables, then we will, like, the end goal is to hopefully be able to replace actually sampling the fish. But for now, I think it's better to use it just in complement to actual sampling.
That's great, Amanda. You're getting a lot of really good feedback. Good job, interesting topic. So I hope you have the chance to go look in there. I have one last question. Do you think that there's a possibility of the salmon populations reestablishing in the Great Lakes based on your study?
I think so, depending on their ability to switch their prey species from the original one that they had been preying upon to this new species that has moved in. There's actually 2 new exotic species that have kind of replaced the original one in the food chain. So I think that's the main dependent variable that'll influence that.
Yeah, for sure. Well, thank you so much, Amanda, and thank you to everyone who presented in this session. Bailey, JC, Jacqueline, and Amanda, this was such an amazing session, and I had such a great day. I hope you all had a great day too, so thank you for joining us.